Thank you for the opportunity to give this talk. And my name is Stephanie Cox. I'm from Gilead Sciences in the Clinical Virology Department. And I'm going to be talking about antiretrovirals for HIV prevention. Or I think I'm going to be talking. Oh, did that just stop it? Oops. I'll try the other one. I just tried that one. That one didn't work. Oh, with this one, sorry. So, back in the late 90s and early 2000s, it was discovered that the same medications that can be used for HIV treatment could also be used for HIV prevention. So we call these pre-exposure prophylaxis, or PrEP. So the first regimen that was tried was Truvada, which is a combination of a nucleoside reverse transcriptase inhibitor, or two nucleoside reverse transcriptase inhibitors, emtricitabine and tenofovir disoproxil fumarate, or FTDF. So Truvada was tried in the IPREX trial, and it was compared against a placebo. The Truvada was taken once a day and showed a 44% decrease in the HIV incidence in this trial. And then the Partners PrEP trial, Truvada was tried once a day, or Viriad, which is TDF by itself, and it showed a 75% decrease with Truvada or a 67% decrease with Viriad. So this was really great data. Seeing prevention for HIV was really a great thing at this time. And Truvada for PrEP was approved in 2012 based on this data. Later studies, the IPERGAY and the PROUD, also showed high efficacy of 86%, really showing the high efficacy of Truvada for PrEP. So in 2015, FTAF, which is emtricitabine tenofovir alafenamide, was approved. And TAF and TDF are two prodrugs of tenofovir. So they have the same mechanism of action, but they have different ways of getting into the cells. So with TAF, you can actually take much less, 25 mg, versus TDF, you would take 300 mg, and you actually get more of the active component, TFE-DP, into the cells, into the PBMC cells. So that was approved in 2015 for treatment, and the next question was, how could we get this approved for PrEP? So we ran the DISCOVER trial, which was the first PrEP trial done at Gilead, and we learned a lot from it. And so we were comparing FTAF to FTDF, and we found at our primary endpoint, which is when 50% of participants had reached week 96, that out of the total population of 5,335 people, we had 7 infections in our FTAF arm for a HIV incidence of 0.16, and we had 15 infections in our FTDF arm for an incidence of 0.34. So this showed that FTAF was non-inferior to FTDF in preventing HIV infection, and we were able to get this approved for prevention in 2019. So while this was great and we were able to get this approved, still out of over 5,000 people, there were only 22 infections total. So these are both really, really great drugs. So in addition, we have real-world data that shows where PrEP uptake is high, HIV incidence declines. And so this is all Truvada data because Descovy or FTAF was not approved at the time. But here in Seattle in 2010 to 2017, we saw a decrease of 42% in the HIV incidence. San Francisco, 2012 to 2016, we saw a decrease of 51%. London, a decrease of 42%, and Sydney, a decrease of 32%, really showing that it's highly efficacious when taken. But not everybody is benefiting from current PrEP options. So people are not good at taking daily orals, they don't like the stigma, they don't like, it's hard to remember, they don't want to carry a pill container around with them. So we really need to find new options for people to make it more prevalent for more people to take. So we have lenacapavir, which is a long-acting HIV capsid inhibitor. And so capsid function is important for several steps in HIV replication: capsid disassembly and nuclear transport, virus production and release, and capsid assembly post-release. So lenacapavir actually acts against all of these and is highly potent. In addition, lenacapavir has long-acting pharmacokinetics, and so a single administration, which is 2 injections, 2 subcutaneous injections, lasts for 6 months. So this has already been tested in clinical trials for treatment, and we would never use it as a monotherapy, so it's paired with something else, but we were also wondering if this could be used for PrEP. And it's been approved in the EU as well as Canada and is currently under review in the FDA. So the first thing we decided to do was try to look at non-human primates and have some proof of concept data to see if a capsid inhibitor actually could be used as a monotherapy for PrEP. So lenacapavir has an analog called GSCA1, and they're very similar. They're novel long-acting HIV/SIV capsid inhibitors. And so you can tell by the structure, very similar, but they're not the same. But they both act against HIV and HIV, HIV being simian HIV, which is what we use. It's a chimeric virus, which we use to infect the monkeys or attempt to infect the monkeys. And so lenacapavir is slightly more potent, but GSCA1 is very potent as well. And we decided to go forward with our proof of concept studies with the GSCA1. So the study design was, it was a repeat mucosal challenge macaque model, and we used Indian rhesus macaques. We had 8 in each group. When we had a group of placebo animals, a group of GSCA1 tested or given 150 mg per kg, as well as a group of GSCA1 given 300 mg per kg. We dosed the animals on day 0 with a single subcutaneous dose, and then we did weekly viral intrarectal challenges. And actually the dose over time went up for the virus. And then we also did infection monitoring, looking at the weekly plasma viral load and serology. So looking at the PK data, a single subcutaneous administration of GSCA1 achieves long-acting exposure in macaques. So we were targeting the IQ4, which is 4 times the protein-adjusted EC95, which is the target for the human clinical dose for treatment. And so you can see here with the lower dose, the 150 mg per kg, that we were able to achieve that for 3 to 10 weeks. And then for the higher dose, we were able to achieve that for 8 to 10 weeks. So here's the results from the study, and GSCA1 did protect from repeat intrarectal HIV challenges. So at the higher dose, we found that 5 out of 8 animals were protected, while at the lower dose, 2 out of 8 animals were protected, while all 100% of the placebo animals were infected within 15 challenges. So this translated to a significant infection risk reduction with GSCA1 versus placebo, 86% versus 96% for the low and high dose. And you can also see that the infections in the GSCA dosed groups occurred only after marked compound washout. So it was later in the time point. This worked really well and gave us the confidence that we could go forward with this as a PrEP agent for human trials. So along the same time, we were starting to think about how we were going to do these human trials because we have some challenges in PrEP trial design now. And so we can no longer do superiority to placebo like we did in those old Truvada trials. It's unethical. We know these drugs work really well, so you can't give people nothing. You have to give them something. And the active controlled non-inferiority superiority trials, that's what the Discover trial was. You can do, but as we saw with the Discover trial, we had over 5,000 people and we had 22 infections. So to really be able to do these, you'd have to multiply that by 6 to 7 times, and that's just a very large N, long timelines. It requires like a certain number of HIV cases. It's not possible in cisgender women either because there have not been enough positive outcomes from the PrEP trials in cisgender women, and so we don't have a non-inferiority margin. So, and that leaves us with the counterfactual background HIV. So what is a counterfactual background HIV? A counterfactual measures what would have happened to participants in the absence of an intervention. So what would have happened if Len for PrEP would not been around? This is not a novel concept. So it's been used in contraceptions for years. And this is actually data that was presented by the FDA, Jeff Murray from the FDA at IAS in 2019, because they're thinking the same thoughts and how can we get things approved when it's becoming so difficult. And so the Pearl Index, what they do for contraceptives is that they actually just have an accepted number of pregnancies that would occur if you were not on contraception. So here it's 80 pregnancies per 100 person years. So then you can actually compare your drug, which you hope to have 1 to 2 pregnancies per 100 person years, and can show that it's highly efficacious. So he suggested we could do the same thing for PrEP. We could find areas that have high incidence of HIV, so like 3.5 per 100 person years, and then we could compare that to our HIV incidence on our study drug to that. And so now that we decided to go forward and do a study that involves this way of doing it, which is called the Purpose 1 study. So the Purpose 1 study is the Len for PrEP in cisgender adolescent girls and young women. It'll be run in South Africa and Uganda. We plan to enroll 5,010 people, which it is actually already enrolling and has been enrolling for the last year. We'll compare LEN to FTAF to FTDF as our internal active control, and then we'll compare to background HIV counterfactual from ARETA for our external control. And that's going to be our cross-sectional HIV incidence cohort to estimate background HIV. In addition, we're also doing the PURPOSE 2 trial. And so this trial is going to be more worldwide. We have sites in several different countries. And this is LEN for PrEP in men who have sex with men, transgender women, transgender men, and non-binary people. So we'll enroll about 3,000 people in this study, and we're going to look at LEN versus FTDF versus, once again, the background HIV counterfactual from ARETA. So how exactly are we going to do this background HIV counterfactual is the next question. Well, we're going to use a recency assay. So this is a tool that can be used to estimate the counterfactual background HIV estimation. And so antibodies have this property that over time, when they are exposed to their antigen, that earlier in infections, they bind less tightly, but over time, they bind more and more tightly. So as you can see in the graph on the left-hand side. And so you can actually just make a cutoff and say anything that binds not as tightly or less than that is a recent infection. Anything that binds more tightly is a long-term infection. And so I have an example of a limiting antigen lag avidity tests. There's multiple different ways you can do recency, but this is one where you have your antigen bound to a 96-well microtiter plate, and then you run the plasma from the participant sample over it. The antibodies bind, and then you use a dissociation buffer that would actually preferentially remove the low avidity IgG antibodies. Then you detect the antibodies, put in a spectrophotometer, get a number, and you have a cutoff that's already preset, that anything below that cutoff is a recent infection, anything above it is a long-term infection. But the next question is, how do you use that data to actually get the background HIV incidence? So we're using a recent infection testing algorithm. And so this is an example. Here we recruit and screen 30 participants. And we conduct standard HIV testing, and 6 of them ended up having HIV. So then we do the recency assay for the people diagnosed with HIV, and we find that 3 are recent infections. So then we can calculate the counterfactual incidence by taking the recent infections over the total number without HIV divided by the recency look-back period. So this is a very simplified equation. Definitely, it's way more complicated than this, but it makes it easier to talk about here. So RETAs have been used by epidemiologists to understand HIV incidence trends at the population level for many years. So this is not a novel concept. This is something that has been accepted. But the PURPOSE trials are the first trials using recency assays, RETAs, to characterize HIV incidence in a screened population as the pre-randomization external control. So this is a novel concept, and we will be the first ones that are trying this. So we're going to see how it works. So I do have a little bit more information about recency in action, because we did the Sienna study. So while we were starting to think about PURPOSE 1 and opening sites in Uganda, we had an investigator tell us that there were 2 sites that she thought would be good places for us to actually use for the study, but they had no background HIV incidence. So we decided to go and use the recency assays in Uganda and try to estimate the background HIV incidence to see if we wanted to enroll them for our study. So we screened 742 eligible adolescent girls and young women aged 16 to 25, and we found that 191 of them were HIV positive. So we did 3 different recency assays: the SETIA lag avidity EIA recency assay, which is the one I described in detail, the Asante rapid recency assay, which is a similar assay but more of a point of care assay, and then the Architect HIV and antigen antibody combo, which is a 4th generation assay but can be used as a 4th generation HIV test that can be used as a recency assay. So we also did viral load testing, because another thing that happens is that sometimes people will come and try to enter trials because there's compensation or they don't want to admit they're HIV positive, and they will be on antiretrovirals. So if this happens, then people usually don't have any virus in their system, and then their antibodies can actually revert back to looking like they're recent. So when you use that in your calculation, you can come up with an elevated HIV incidence number based on the amount of people that are actually on ART. So what they do to try to eliminate this is actually use a viral load cutoff. And so our viral load cutoff was 75 copies per mil. And so once you eliminate those people, then hopefully you can get the actual real numbers. And so out of our 191 HIV positive participants, we had 44 that were less than 75, so a sizable amount. And so then we were left with the 147 that we could actually evaluate the recency data and determine what the background HIV incidence was. So reader results by site, and so we had 2 sites, Hoima and Mitiane Mubiende. And for Hoima, looking at the lag EIA, with for the recent samples, we had about 15% that were considered recent, and then in Mitiane Mubiende, we had 21% that were considered recent. For the Asante, we had 9% that were considered recent versus 12% in Mitiane Mubiende, and the Architect, 11% versus 15%. And that translated to a background HIV incidence of 3.1 for Hoima with the lag EIA, 1.9 with the Asante, and 1.6 with the Architect, compared to the Mitiane Mubiende with 23.2, 14.4, or 14.3. So based on this data, we did choose Mitiane Mubiende to be one of the sites in our Purpose 1 trials, and we decided not to use the Hoima site. So in conclusion, oral PrEP, FTDF and FTAF is highly efficacious when taken daily, but adherence can be an issue. Lenacapavir is a 6-month injectable that is currently under study for PrEP, and proof of concept studies with the Lin analog GSCA1 demonstrated high efficacy as a PrEP agent in NHP studies, which gave us confidence to go forward. And due to challenges with running PrEP studies, new study designs are required to evaluate new PrEP agents in development. So our phase 3 studies, the Purpose studies, are currently being run with Len for PrEP using a recency assay to determine background HIV incidence. And using a recency assay to determine background HIV incidence was demonstrated by the Sienna study, providing data to enroll a clinical site. So I want to acknowledge all my colleagues at Gilead Sciences who've done so much of this work, as well as our collaborators at Beth Israel Deaconess Medical Center who were involved in the NHP studies, the Muju Care, Muju Research Collaboration who did the Sienna study, recency assay experts who helped us choose recency assays and discussed and just gave us overall knowledge, as well as the participants, their families, and all participating investigators. And thank you for your attention, and I'll take any questions.
Hi, it was a great talk. Do you know the half-life of lenacapavir?
I don't actually know the half-life of, because I mean, it's 6 months, but I don't know what.
Okay.
Yeah, sorry. Okay. Christian, do you know?
It's in weeks.
Okay, thank you. Also, have you looked at any off-target effects of this small molecule?
Of lenacapavir?
Yeah.
I mean, it's already been done in clinical trials, and their safety profile is really good, so there are no clinical safety effects that we can see. Thanks.
Very nice talk, thank you. The resistance profile for lenacapavir is quite complex. And have you looked in those populations, particularly the Ugandan population with the high recent incidence for pre-existing resistance?
So we've done some and looked there, and we don't see anything that we can find. So, it's not super extensive, but we expect, because we've looked at all the databases, and while they're not, I think there's a decent amount of samples from South Africa, which is the majority of where our Purpose 1 study is, and subtype B is the other subtype for the Purpose 2 studies, which is the majority, and we haven't seen anything as of yet.
Okay, thank you.
This is a question about human behavior. So it might be harder for me to remember something I had to do every 6 months than something I had to do every day. So what is the optimal dosage of something to achieve compliance?
So I think that's really interesting, and I have a background in contraceptives as well, and so, I think 6 months, especially, we are thinking you can take it at home, but that's why in some ways I feel like going to your doctor actually wouldn't be a horrible thing, because if you make the appointment, that makes it easier to remember. Daily is hard. I think anyone who's taken birth controls or any other kind of daily medicine, it's hard to remember what you did the day before. We're also working on weekly and monthly, but I kind of feel like the same things could happen. So I think it's really going to be person dependent, and whether they put it in their calendar or how it works, it's going to be interesting to see, because You know, I do think there's something to be said to go to the doctor, get an HIV test, and, you know, maybe get it done at that time. And once every 6 months is not asking too much versus if you had to go every month or every week and have to get something.
So, I mean, I'm just wondering about compliances. I mean, you know, like getting your teeth cleaned, right? People, they nag you until you show up. But that's here, you know? I
Yeah.
don't know how that would be in a more impoverished setting if the clinic contacted you and said, you need to come in. Is that infrastructure there? Is that the goal?
And it depends on where it is, but I do think actually the infrastructure is like in South Africa and things. I mean, in some ways it's better than the U.S., I feel like. So, you know, I think it will be interesting to see how it all works out, but I think it is there, and this would be a game changer, hopefully, for people.
For the recency testing, did you test for ART drug levels in any of those?
No, we did not, which actually one of the investigators did say that she thought that there was an enormous amount of elite controllers in Uganda, so we're thinking about testing for ART levels, because I can't imagine there's that many.
Yeah.
I just have to believe that there's a lot of people on ART, but we have not done any testing so far, though I have the samples and we were considering doing that.
Yeah, we've seen that in the blood bank in South Africa. We have all these elite controllers who are donating blood, and then we find out that, you know, 80% of them have drugs in their system, so.
Well, I mean, that's what you would expect, but she seemed to say that she did think there was some study that said, and so I said, well, that's fascinating in itself. If Uganda has a lot of elite controllers running around, I'd like to know that too.
I was curious related to the compliance question. If there are risks associated with monotherapy in terms of antiviral resistance mutants that arise if, depending on the level of compliance and whether somebody's sporadically taking it and then becomes HIV positive and don't realize it and then continue to sporadically take it?
I mean, so there are risks involved. I mean, hopefully we would get people on a full regimen if they were to go off of it. You know, if someone was to say, I no longer want to get these injections, then we would want them to be on a full regimen so that they would not get resistance to LEN. One thing that is nice is that, you know, the approved regimens all have INSTI based, and so even if you were to get resistance to LEN, which is not what we would want, but you could still go on the major regimens for HIV treatment.
Since we're a minute ahead of schedule, I also had a question. For the Purpose 1 trial, are you excluding women on hormonal contraceptives, or will you be looking at that and how it might affect the PrEP efficacy?
So it shouldn't affect the PrEP efficacy at all. We're not excluding them, so they will be on there. And I mean, you know, we think of different ways that we could even combine these different things and things like that. So yes, we'll have all that data, as well as transgender women and looking at, you know, different hormonal supplements that they're taking and things like that as well. Thank you.
Thank you.